Hello friends, welcome back to another video from Shomus Biology. In this lecture, we are going to talk about the cell cycle regulation. In this series of lecture about cell cycle and cell cycle regulation, we are discussing about different aspects of cell cycle regulation. We have talked about the cell cycle phases and checkpoints. We have talked about the role of cyclins and CDKs in cell cycle. Now in this the ending video, we are going to discussing about the cell cycle regulation. And basically, first I'll discuss about how the cell cycle is regulated as an overview. Then I'll go to the actual process of cell cycle regulation. We know one thing till this point, that in the cell cycle regulation, there are proteins involved, specific proteins. And we have talked about specific proteins. Proteins like cyclins, right? We have talked about proteins like CDK. And we know cyclin and CDK work together as a partner in order to regulate the progression of cell cycle from one phase to the other. And we know for the progression of G1 to S, there is one type of cyclin CDK partner. From S to G2, there is one type of cyclin CDK partner. For G2 to M, there is another type of cyclin CDK partner. Now the question is how exactly these cyclin and CDKs are regulated. We also have understood the importance of cell cycle checkpoints. If there is any error, any kind of DNA damage, or if the cell lacks the volume of the cytosol and change in the cytosol nucleus ratio, in that case the cell cycle progression may be stopped, known as cell cycle arrest, which will be done by specific proteins. Again, cell cycle arrest will be done. Now the question is how exactly all these things are correlated, all these things are connected, right? That's what we need to know. बीच बीच में हिंदी में कुछ कॉम्प्लिकेटेड टॉपिक हो तो वो समझाएंगे. पूरा वीडियो जो है इंग्लिश में ही होगा. So the thing right now we are trying to figure out is basically the process of the cell cycle regulation. So till this point we talked about a protein, mainly an enzyme, kinase. A kinase enzyme's job is to add a phosphate group, right? Phosphate group to a target molecule, and once adding the phosphate group to the target molecule gets activated. Generally, phosphorylation causes activation. And that activation triggers next series of events. But once we add phosphate group to activate something, we should have something to inactivate those same molecules, right? Because desensitization is a crucial and important step during any sort of cell signaling or signal transduction pathway that goes on inside a cell. So in this case, what we simply know is that the CDK is an activator. So obviously we need some other proteins which actually help the cell to progress from one cycle phase to the next phase, as well as they can act as an inhibitor. So what is acting as activator or inhibitor here, apart from kinases? Okay, so these are the first kinases. Apart from kinases, we have some more, right? And this is what we are going to talk about, and they are also behaving like a kinase protein, because kinase protein always add phosphate group. So kinases, we know that CDK is the major enzyme for the changes that is needed for the cell cycle progression. But along with that, what else we need? We also going to need two more type of kinase here. One is known as V1 kinase and another one is known as in this case CAK. CAK. It's also known as CAK kinase or CAK, like this. CDK activating kinase. And for desensitization events, we also need phosphatase. Okay. So apart from that, we need this phosphatase. An example of this is CDC25, for example. CDC25 is a type of phosphatase which will do the job of desensitization. Means basically it will cleave the phosphate group out from all those active proteins and will make them inactive. This activation of proteins and inactivation proteins both are equally necessary in order to progression of the cell from one phase to the next phase. Now we'll move on to the actual mechanism of the cell cycle regulation. And to understand the actual mechanism, I read from multiple books, and students always ask me that this particular part is very complicated. The graduate students, the graduation of microbiology, biochemistry, molecular biology and biotechnology students always find it confusing. But after this lecture, it will no longer be confusing. सिर्फ एक ही चीज़ को याद रखना है. ओके, एक सिंपल सी लाइन है, इट्स अ निमोनिक ट्रिक है. इसको अगर ध्यान में रखना है तो ध्यान में रखने के बाद you can answer any question from this and you will never forget the cell cycle regulation. और वो line जो है, so what is that particular line? We try to catch polo. Polo is someone, or let's say somebody, but we try to catch polo. If you remember this particular, to catch the polo, let's say like this, we try to catch the polo, it will make it even better. We try to catch the polo. This would be the line that you should remember. Simply ये line को याद रखना है. क्यों इसको याद रखना है, क्या काम आएगा, अगले स्लाइड में देखते हैं. But this is the mnemonic to remember, this is the trick to remember. We try to catch the polo. If you remember this, you will be solving, you will be remembering all this topic throughout your life. So the first thing that I want to tell now, the process of cell cycle regulation. How exactly cell cycle regulation is done. So here, remember that same line, we try to catch the polo. This is what we need to remember. These are all the important words of the line. Now why they are important, I am coming here in a moment. But remember that we have discussed about the importance of kinases. We have discussed about the importance of phosphatases. And the kinase working here is V1. The kinase working here is CAC. Okay. And the phosphatase working here is CDC25. By this line will give you information about all this. V will give you V1. Alright. So tyrosine. Okay, tyrosine 15. What is tyrosine 15? Why I am writing this? I will be explaining everything to you, so don't worry about it. To catch the polo. C, catch is go for CAC. Okay. The TH threonine 161. Okay, polo is only polo. So these are the important components that we are going to utilize while understanding the process of cell cycle regulation. We have V1, tyrosine 15, CAC, threonine 161, and we have polo. Apart from all this, only one thing was missing in this particular line, that is the role of CDC25, which we will see in a moment because that is phosphatase, so we keep it separated. So now we are going to see the actual process, and for that I am going to change the color. I will take red color again, and we are going to discuss about this whole process. Now remember one thing, that let's imagine that we are talking about M phase, because the regulation of cell cycle occurs in individual phases, because before moving from one phase to the next phase there is a checkpoint. So based on the checkpoints signaling, the cell cycle progression is actually done. So let's imagine the M phase. We are talking about M phase progression. So what we are looking at is, we have M phase CDK, M phase cyclin. And if you recall, the M phase cyclin is B cyclin or cyclin B plus CDK partner is CDK1. How to remember which cyclin and CDK partners are involved in a particular phase of cell cycle? For that, you need to watch the other video of mine regarding the cyclins and CDKs of the cell cycle regulation. वो वीडियो देख लेंगे तो एक बार टोटल क्लियर हो जाएगा कि कौन कौन सी CDK cyclins इन्वॉल्व है, क्या काम करता है, सब कुछ. So now we are going to talk about the process of regulation. So in this case, let's imagine the same thing. We have this CDK. CDK is the main partner. So this is CDK1, the M phase CDK. You can also refer to as M phase CDK. But this CDK is non-functional without its cyclin partner. So I'll take another color to describe the cyclin partner here. And let's say this is the cyclin partner, that is cyclin B. So CDK and cyclin B are there together. And once cyclin B associate with CDK, CDK is active. So this CDK is in active form. No. Now till this point, when we discussed about all this idea that CDK is the actual enzyme, once CDK is active it will phosphorylate other because it's a kinase. Everything is fine. And I told you that CDK once associated with cyclin, it will be active. Fine, that's also active in terms of very crude level. But now you dig deep. And what we understood from here is that CDK, whenever associated with cyclin B, it can now be ready to be activated. It's ready to be activated, but still it's not active. CDK is a very crucial enzyme inside of the cell. It's a kinase. It can phosphorylate many other proteins that can lead to the cellular growth, cell division. And if it's misregulated, it can turn a normal cell into a cancer cell. Because tumor can form and then it can become malignant and can get cancerous. That's why CDK is very tightly regulated. It's never be always active. Although it's associated with cyclin, it can be activated, but it's not active yet. It is yet to be activated. मतलब अभी तक activate नहीं हुआ है, activate हो सकता है. Now for the activation part, why the CDK1 is not activated? There is a reason behind it. Because the CDK, once associated with cyclin, there are two places of the CDK. One is this tyrosine 15 and another one is this threonine 161. Tyrosine 15 and threonine 161, both are there. Now what are their functions, what are their role? Let me explain that. I'll again change the color. It's very important to change the color because otherwise you will not understand. I'll take the black color in this case, in this instance. Okay. So the thing is, in this case, the first phosphorylation. So there are two phosphorylation. First phosphorylation would be done in tyrosine 15 by V1. V1 is again kinase that can add a phosphate group. It is adding a phosphate group to the tyrosine 15, means the residue number 15 of the tyrosine amino acid residue. It attaches the phosphate group. And this phosphorylation is known as inhibitory phosphorylation because it prevents the activation of CDK. Sorry, it inhibits the phosphorylation of activation of CDK. So it acts as an inhibitory phosphorylation for the CDK. Then the second phosphorylation is done. So this phosphorylation is done. There is a phosphate group, but the CDK is not yet active because this is inhibitory phosphate. In order to make CDK1 active, this phosphate needs to cleave out. But the phosphate is in on place, so CDK to activate abhi tak nahi hua. Now the next step is another enzyme and another second phosphorylation, and this second phosphorylation is done at the threonine 161 residue by CAC. Another kinase phosphorylates threonine 161 residue. Second phosphorylation. This is activatory phosphorylation. But once the CDK is with these two phosphorylations, still it will be inactive. You may ask a question, but why? Because the inhibitory phosphorylation is still in the place. This is activated. Okay? Inhibitory is still in the place, so the CDK is still not active. When the CDK will be activated? Once the inhibitory phosphate is cleaved and the CDK is with only activatory phosphate, then only the CDK will be activated. Okay? Now, for that what we need to do? We need to cleave the inhibitory phosphate out, isn't it? Now you see the importance of phosphatase enzyme, desensitization. Phosphatase enzyme's role. So in this case, phosphatase CDC25. I'll take a different color here. Red one again. Now CDC25 will cleave the inhibitory phosphate out and will make CDK with only one phosphate to the active site, that is threonine 161. THR 161. Okay, this is where the phosphorylation remains. The activatory phosphate is in the place. Now the CDK along with the cyclin, obviously cyclin is there. Here also the cyclin is present. The CDK gets active. This CDK will now ready to phosphorylate other proteins. Phosphorylate other proteins necessary for the movement of the migration of one particular phase to the next phase of the cell cycle. This is how the whole process is done. For M phase movement, this is how it's done. Okay. So try to understand this situation. Now try to correlate with the line that we said. We try to catch the polo. V1 phosphate in that order, in that sequence. V1 phosphate works the first, V1 kinase acts as a first phosphate addition to the tyrosine 15 residue. Then comes CAC, second phosphorylation by CAC kinase to the threonine 161 residue. Then the last thing is basically CDC25. But CDC25 is a phosphatase which is activated by polo kinase or polo-like kinase known as PLK. Polo-like kinase protein. That's why you keep polo here, not CDC. So we try to catch the polo. V1 tyrosine 15, catch CAC threonine 161, polo activates CDC25. Rest of the process is crystal clear. I rest my case. That's how easy it is to understand. हिंदी में एक बार summary. नॉर्मली CDK एंड साइक्लिन पार्टनर जो है, जब भी साइक्लिन के साथ CDK होता है वो एक्टिवेशन के लिए रेडी होता है मगर एक्टिवेट नहीं होता. फर्स्ट V1 टायरोसिन 15 रेसिड्यू पे फॉस्फेट ऐड करता है मगर वो इनहिबिटरी फॉस्फोराइलेशन है, वहां पे भी एक्टिवेशन नहीं होगा. सेकेंड फॉस्फेट कैक काइनेस थ्रेओनिन 161 पे ऐड करता है, वो एक्टिवेटरी है मगर जब तक टायरोसिन 15 पे V1 फॉस्फोराइलेशन है तब तक ये एक्टिवेट नहीं होगा. दो फॉस्फेट जब तक रहेगा फिर भी एक्टिवेट नहीं होगा. देन कम्स द थर्ड इंपॉर्टेंट एंजाइम हियर, पोलोकाइनेज. पोलोकाइनेज एक्टिवेट करेगा CDC25 को. CDC25 इज एसेंशियली अ फॉस्फेटेज जो कि फॉस्फेट ग्रुप को क्लीव करता है, निकाल देता है. तो फॉस्फेट ग्रुप कहाँ का, इनहिबिटरी फॉस्फेट को निकाल देगा. दैट कीप्स CDK के साथ सिर्फ एक फॉस्फेट को एक्टिवेट रखेगा जो कि थ्रेओनिन 161 पे है. तो थ्रेओनिन 161 का फॉस्फेट विद CDK, नाउ CDK विल बी एक्टिव. अब ये CDK जब एक्टिव हो जाएगा ये इसका काम करेगा. That's how easy it is. Now the final thought I'll move into, so you can take a screenshot at this moment. I believe it's a clear view now. Done. Let's move to the next part. The last thing that we are going to talk about, the loop, very very important. So give you a revision that this is the CDK, obviously cyclin are there. I'm not writing it here every time. So the first so this is normal CDK. I must not, so it's a normal, let's say draw with normal CDK here with cyclin partner, gets first phosphorylation and that is done by V1. Okay, V1. Then the second phosphorylation by CAC, right? This is the first phosphorylation, this is the second phosphorylation. First one is inhibitory, second one is activatory. Then the last step, CDC25 cleaves this phosphate out. So CDK, this phosphate is no longer here, only one activator phosphate. This is the active form of CDK. This was inactive form. This was also inactive form. And this is the active form of CDK at the end. Now what else we need to understand in this particular very easy to understand sequence of events is the role of the feedback loop. Now what is the feedback loop? Feedback मतलब जैसे feedback होता है, मैं कोई सर्विस आपको प्रोवाइड कर रहा हूँ, आपको अच्छा लगा तो आप लाइक करोगे, दैट्स अ फीडबैक. अच्छा नहीं लगा डिसलाइक करोगे, दैट इज अनदर फीडबैक. कमेंट करेंगे तो दैट्स अ फीडबैक. सो ये ऐसा फीडबैक होता है हमारे लिए. सो यहाँ पे भी जो फीडबैक है दैट्स अ फीडबैक लूप. हाउ इट्स डन बेसिकली, once the CDK gets active, then this active form of CDK and cyclin, obviously cyclin is there throughout this place, cyclins are there. Okay, you must draw it correctly. So cyclin is there in every single place. So once the CDK is active, this active form of CDK has two set of positive feedback loop. What are the positive feedback loop? One is this is going to active and signal basically to the polo. Polo activates CDC25, right? So जब भी CDK activated form में चला जाएगा वो पोलो को इन्फ्लुएंस करेगा. फर्दर पोलो तैयार होगा तो CDC25 भी एक्टिवेट रहेगा. CDC25 का काम होगा फिर से एक्टिवेट करना CDK को. So that's a positive feedback loop, right? So it's actually CDK once active try to increase its own concentration by this manner. Multiple activation. A positive feedback loop is initiated. Similarly, this active CDK will inhibit V1. So this is the inhibition of V1. V1 itself is inhibitory, right? V1 adds the inhibitory phosphate. So now if CDK, the active CDK inhibits V1, that means what? That means there is no V1. So automatically from the first point of time, CDK will get only one phosphate, that is the activator phosphate, and the CDK will be activated. These two feedback. This is also positive feedback. Why it is positive? You may think like, why not is negative? Because ultimately the resulting behavior is to the positive side, that is making more CDK, making more active form of CDK. And both the case, either activating polo or deactivating V1 help the cell to achieve this goal. That's why it is called as a positive feedback loop. So both are positive feedback loop. But what is the requirement of V1? Most of the students are baffled, and when I was a student I was baffled by this idea. Why cell is doing so stupid things, first adding an inhibitory phosphate, then adding an activator phosphate. Why not use only activator phosphate? What is the necessity of V1? Don't you think this is logical question? This is absolutely logical question. Now there is a serious answer to that. That is the V1's role is actually, V1 gets activated by whom? By the checkpoint kinases. What are checkpoint kinases? Checkpoint kinases are specific enzymes which can detect the DNA damage during the process of cell division. So कभी भी कहाँ कोई भी DNA डैमेज होता है, सिंगल स्ट्रैंड, डबल स्ट्रैंडेड, उसको डिटेक्ट करेगा V1 जब उसको डिटेक्ट करेगा, so V1 को इसीलिए रिक्रूट किया जाता है. V1 is recruited by the checkpoint, so depending upon the situation it will be recruited. Once V1 is recruited, this V1 will then go, and V1's job is to inhibit the movement of cell cycle. That's why it's an inhibitory kinase. But in a cell, whenever we need the cell to grow, we need a lot of activated form of CDK. So how can we produce so much concentration of activated form of CDK in less time? The only way it's possible is to fuel it up, means to make it active already, but also to block somewhere like inhibitory phosphate. So 2-phosphate is the situation where the CDK is almost ready, simple requirement of phosphatase activity to make it active. Because bringing the ATP, transferring of the phosphate for the kinase activity is more difficult than cleaving the phosphate out, right? That's why the cell always prepares the CDK. CDK always present, let's say once a single moment the CDK concentration is high, but all are biphosphorylated. 2-phosphorylation is done, so they are inactive. The moment CDC concentration will rise due to the polo activation or polo-like protein activation, polo-like kinase activation, it will cause cleaving of all the inhibitory phosphates. So we will get all the active form of CDK at a given moment of time. It's very important for the cell division to occur. Concentration of CDK, active CDK, actually regulates when the cell cycle will continue and progress to the next step. That's another important signal that the cell requires. So it's like that we are all running for the race, and we want to prepare our car or bike, whatever you take for the race. But if we don't have petrol in it, what we should do, we'll go to the petrol pump, fill the petrol and then move. Before that, we fill our tank, all the bikes, but we keep them, we take their keys out. So they are ready. Once started, they will go fire. But they can only do if we provide them the key. चाबी देंगे तभी वो स्टार्ट करेगा, तभी वो निकल पाएगा. टैंक तो फुल भरा हुआ है. अगर टैंक खाली है, चाबी भी दे दोगे तो जाएगा, बढ़ाएगा, टाइम लगेगा. तो एट ए टाइम हमको बहुत सारे राइडर्स एक साथ नहीं मिलेगा. तो बहुत सारे राइडर्स को एक एक टाइम में चाहिए, तो आपको हर एक राइडर्स की जो टैंक है उसको फुल कराना है, चाबी लेके रखना है, सिर्फ चाबी दे देना है, बस वो स्टार्ट हो जाएगा. दैट इज द आइडिया. दैट इज द आइडिया बिहाइंड V1 and CAC mediated phosphorylation and the role. That's the overall idea about cell cycle regulation. I believe the cell cycle regulation is crystal clear now. Don't get confused, and happy studying. If you like this video, please hit the like button, share this video with your friends and colleagues, and subscribe to this channel to get more informative videos like this, and watch this whole series of cell cycle regulation lectures. You'll be greatly benefited. Thank you. Bye.